Hello everyone, hope you are all fine and doing well. Today we will see about inflammatory bowel disease. Inflammatory bowel disease is a chronic condition that results from complex interactions between intestinal microflora, host immunity in genetically predisposed individuals. So it includes a conglomeration of factors: intestinal microbiome alteration, host immunal activation in genetically susceptible individuals. So all these factors, they lead to inappropriate mucosal immune activation. This includes two entities: Crohn's disease and ulcerative colitis. This is ulcerative colitis, this is Crohn's disease. Ulcerative colitis involves left side of the colon, the rectal and the sigmoid region, whereas the Crohn's disease is more common in the terminal ileum. This produces continuous lesions, whereas Crohn's produces patchy lesions in between normal intestinal mucosa. So it is characteristically described as skip lesions. Both the inflammatory bowel disease are more common in younger age group. In teenage, early 20s, this is more common. And there is also a racial predilection which is more common in Caucasians and the Americans, and also in Ashkenazi Jews. It is also seen in increasingly prevalence in North America, Northern Europe and Australia. But of late, over the past 1 or 2 decades, the epidemiological factors play a role and it is seen in increasingly incidence throughout the world. So what are the reasons for increasing incidence of inflammatory bowel disease around the world? Because of improved food storage conditions like invention of refrigerators and other methods of storage of food, decreased food contamination, and preservatives and added materials to processed foods have increased. And main etiology is changes in gut microbiome composition. That is, the gut microbial flora is altered. There are inadequate regulatory processes which will limit mucosal immune responses, and that leads to increased inflammation. These mucosa-associated microbes, they trigger persistent and chronic inflammation in genetically susceptible hosts. Genetic factors, immunological factors and gut microbiome. These three play an important role. Now coming on to genetic factors, NOD2 gene polymorphisms play a very important role, more so in Crohn's disease. There are also abnormal mutations which are diagnosed in immune signaling and immune regulatory genes like interferon gamma, interleukin 10 and its receptor. There is also activation of T helper cell 2 and T helper cell 17 signaling activation mutations. Both Crohn's and ulcerative colitis also have increased susceptibility in abnormalities in interleukin 23 receptors. So, because of all these immune activation mechanisms, there are associations of these inflammatory bowel diseases with other autoimmune diseases like insulin-dependent diabetes mellitus, rheumatoid arthritis, psoriasis and other conditions. So, this pathogenesis, this NOD2 gene polymorphisms, if it is very much susceptible, then any ileal disease will be having an aggressive impact. Any bacterial infection, the normal epithelial, it will undergo injury and it will produce triggering of the activation of gut associated lymphoid tissues, producing mucosal inflammatory response. And ileal disease, it's more common and it will have more aggressive nature if the patient has NOD2 gene polymorphisms. This infection with susceptibility with NOD2 gene polymorphisms increases the mucosal bacteria, and the normal bacterial flora, the Faecalibacterium species and other things are decreased, whereas increase in Escherichia species. So, all these will lead to abnormal and pathogenic commensal bacteria that leads to mucosal immune activation and triggering to development of inflammatory bowel diseases. Apart from this NOD2 gene polymorphisms, we also have abnormality in autophagy and cellular stress response genes, which are involved in autophagosome formation. So, you would have read previously in the general pathology itself what is an autophagosome. So, if any foreign body invades, the normal immunity, it will try to engulf that and destroy that. So, these genes are concerned with autophagosome formation and destruction of the microbe or any other foreign body. This abnormality in these genes ATG16L1 and IRGM may lead on to inflammatory bowel disease. Also, we know about the Paneth cells which are present in the intestine. They may secrete certain antibacterial peptides which are not normal but abnormal that may trigger the mucosal immune system.